Welcome to class. So let's look at ways of control. So how do we control these disease-causing organisms? How do we go about it? So, control of disease-causing organisms. Control. Yeah, so control of disease-causing organisms. Now, one way of controlling disease-causing organisms is by use of antibiotics. Use of antibiotics. Now, antibiotics work against infections caused by bacteria. So when we see disease conditions resulting from bacterial infection, one of the things we recommend that such a person should take is antibiotics. Now, what is the goal of antibiotics there? Antibiotics, we have different forms. We have some antibiotics that can kill the bacteria. So we refer to those antibiotics as bactericidal. So antibiotics that succeed in killing the bacteria, so antibiotics that kill the bacteria, are called bactericidal. So such antibiotics are called bactericidal. So we refer to such antibiotics as bactericidal. While those that inhibit, they limit the growth of the bacteria by inhibiting protein synthesis, are bacteriostatic. So antibiotics that inhibit protein synthesis, that's how they limit growth, inhibits protein synthesis. Those that inhibit protein synthesis are called bacteriostatic. Are called bacteriostatic. So they are called bacteriostatic. While those that kill the bacteria are called bactericidal. Those that inhibit protein synthesis are called bacteriostatic. A good example of bactericidal is your penicillin. So bactericidal is your penicillin, while bacteriostatic is your tetracycline. And these are commonly used antibiotics. Your tetracycline. Now you can also use antiseptic. Like in the olden days, before the discovery of antiseptic, most of the soldiers that go for war, they eventually die from injury, abrasions, and cut wounds. Those wounds which they sustain, those abrasions, those gun wounds. So they end up dying from them before antiseptics were discovered. So the goal of antiseptic is that it prevents the entry of these bacteria into the tissues. Basically, they are bactericidal actually, in a sense. So when you apply it, and that's why if you want to dress wounds, you apply antiseptic there. The idea is you want to kill whatever bacteria will be on the surface of the skin. You don't want it to enter into the skin. So when you see wound, you apply it, you kill it off, then you dress it. So once this was discovered, it saved a lot of lives of soldiers. So soldiers are no longer dying from cut wound. Because what was actually causing them to die is not the wound itself, it's not the gun wound itself. It's actually due to the fact that their skin is exposed and you have this bacteria seeping in, cause to come down with sepsis. And sepsis is one of the quickest ways to die, where you have so much bacteria in your blood and it brings about inflammatory reaction. So bacteria plus inflammatory reaction equals to sepsis, sepsis equals to death. And also use of high temperature. The famous scientist, the French scientist Louis Pasteur, was the one who demonstrated that at certain temperature, sufficiently high temperature, bacteria will become inactive and they can die. Even then, further studies now show that bacteria actually work best at a temperature of, so the optimum temperature for bacterial activity is 37 degrees Celsius, and that's more like the body temperature. So the optimal temperature for bacteria is 37. Now you find out that temperature below that, you see that the bacteria will be rendered inactive. And that's the phenomenon you use in your deep freeze compartment, because at -4 degrees Celsius. That's why fresh food are stored there, to prevent it from going sour. Your milk, your fresh is kept there to prevent it from spoiling. And also you see this applied even in cooking. You heat your food to sufficiently high temperature. Why? Because you want to keep whatever germs could be residing in the food. So it's also applied in surgical procedures. After all surgical experiments have been done, they put in a machine called the autoclave. Autoclave is a machine that heats up those surgical equipment to some extremely high temperature. The idea is, at that temperature, every microorganism, every disease pathogen present will be dead. You also do dehydration. Dehydration is also another means, because when there's unavailability of water, the microorganism will not be able to survive. So dehydrating is another way of preservation. You use it for fishes a lot. That's one way of preserving fish, dry fish. So it's one way of preserving fish. So dehydration. So when you dehydrate food supplies, you are going to prevent the growth of microorganisms, because there's no water, no oxygen for them, and they just die off. So in next class, we still have more ways of controlling microorganisms.